Okay, so our next chapter is going to be about what to do with nitrogen when you are using your carbons for something else. And nitrogen is going to have to be dealt with, and it's dealt with primarily through your urea cycle. And so again, your ammonia is going to be toxic, so you want to prevent that from forming. In your diet, as we just talked about, you're going to have amino acids that are going to be released. Your amino acids are going to be picked up, and these can be used to synthesize proteins that are necessary for a cell. Your carbons that are extra, or your amino acids that are extra, if you have a high protein diet, for example, those can be used for energy, also made fats, etc. And the nitrogen is going to be passed on to urea. The fasting, your muscle is going to recycle proteins, convert them to amino acids, and then be able to use those for energy. So your flow of nitrogen, just in general, you have sources of nitrogen, and those sources are going to be external. So you have your bacteria, and bacteria have the ability to fix nitrogen. Plants are going to have nitrogen. You, as an animal, we are humans, so we are going to take up nitrogen in the form of proteins, amino acids, and then we're going to digest them. We're going to make our own proteins, of course. In animals and us, glutamate is going to be important as a nitrogen acceptor and a donor, and this is through two different enzymes, glutamate dehydrogenase and glutamine synthase. So what are the fates of amino acids? You kind of know these fates. We're going to be able to generate energy. So we can generate energy from the carbon skeletons. We also have free nitrogen, and this can go into the urea cycle. We can also use free nitrogen to make amino acids, nucleotides, etc. We have a cycle in between that connects your citric acid TCA cycle to your urea cycle, and this is a shunt. This is an aspartate arginosuccinate shunt, and it uses it will feed back and forth between the citric acid cycle to the urea cycle. And then, of course, we can also take carbon skeletons from our proteins, and we can generate glucose through gluconeogenesis. So just a general overview of the urea cycle. So the urea cycle, in which you have your carbon, nitrogen. Your carbon can be used for energy, can be stored, glycogen, fatty acids. So carbon sources, we can go for storage. We can also go for energy. When those carbon sources are going to come from proteins, again, you can have your carbon sources, and then your nitrogen is going to have to go to the liver for urea. You're going to, in your urea cycle, you're going to get one nitrogen from ammonium. So there's one source of nitrogen. And then you're going to have a second nitrogen that's going to be added, and that's going to always come from aspartate. So your two nitrogens are going to form urea. So this is just a picture about amino acid metabolism, and there's two different versions. There's fed and fasting state. In a fed state, as we've talked about already, you're going to digest your proteins, start in the stomach with pepsin. It's going to go into your gut, and you're going to have different enzymes, trypsin, chymotrypsin, elastin, etc. Break them down into amino acids or peptides. Amino acids or peptides can be absorbed. Your amino acids are going to go in through a sodium co-transport. And your peptides are a hydrogen co-transport. And then those are going to be broken up into amino acids that get then released. Amino acids are going to be picked up by your liver or by other cells. primarily used to build proteins in other cells. In your liver, you can do many different things. You can make glucose, go to glycogen stores, you can make fats, VLDL particles, etc. It just depends how much excess proteins you take in. Now, in your fasting state, this is when you're going to have to mobilize carbons in order to use as fuel because you don't have it coming in through digestive. And so in your fasting state, you are going to, again, have your liver. You're going to have your muscle cells. Muscle cells will take proteins and break them into amino acids and generate energy. They will also release their nitrogens in either glutamine or alanine. Glutamine is going to go to the gut, or glutamine is going to go to the kidney. Both the kidney and the gut will transfer those nitrogens back into alanine, and alanine will feed into the liver, and it's going to go towards gluconeogenesis, or ketone body formation. The nitrogen gets packed up into urea, and it goes into urine for excretion. So the glutamate dehydrogenase reaction is important, and it's going to depend on your ATP and ADP levels. So you have your ammonium ion, and you have alpha-ketoglutarate. And this is going to, in the presence of NADPH, is going to make glutamate. And this is going to require glutamate dehydrogenase. This is going to use ATP or GTP. Okay. Now, glutamate can also go in the reverse direction, of course, and this is also glutamate dehydrogenase. And this is when you have low ATP or GTP. Now, glutamate dehydrogenase type 1 is going to be in all tissues, and glutamate dehydrogenase type 2 is going to be specific for the brain. Okay, so you also have glutamate, and this was made from your previous, right? That was the dehydrogenase reaction. So I'll call this number 1. Okay, so our glutamate, shown here, is what we're picking up with over here. So that's the first reaction. And so you have one nitrogen associated with glutamate, and then you're going to again need ATP through glutamine synthase. Synthetase. And then this is going to become your gamma glutanol phosphate. And then you're going to have another nitrogen come in, and you're going to release your inorganic phosphate. And this is, again, through your glutamine synthetase. It's like 2 reactions, same enzyme. And you're going to get glutamine. And this has 2 nitrogens. Now in the kidney and the brain, you're going to take your glutamine, also the gut, and they have glutaminase. And this is going to release the nitrogen, and in the kidney it will buffer. You're going to get glutamate. or this can be passed on to alanine. Now, glutaminase, sorry, should be kidney. Glutaminase in the brain is going to synthesize glutamate. And in the kidney, it's going to be an acid-base balance. So it's a buffer. Now, in the liver, you're going to be able to express both glutamine synthetase and glutaminase. However, this does not occur in the same hepatocyte. So you have different hepatocytes. This is going to make sure that the liver is not both a producer and consumer of glutamine. And it allows your liver to scavenge ammonium and not incorporate it always into urea, because ammonia the nitrogens can be used to make amino acids. So enzymes of the urea cycle are in hepatocytes that are going to express glutaminase. So what that means is in your liver, not all hepatocytes are going to be actively participating in the urea cycle. So in cases when you have acidosis, this is when more glutamine is diverted from the liver to the kidney. And this is to conserve bicarbonate, which is sort of a buffer that will lower the pH, will increase the pH, sorry, make it more basic, because bicarbonate is necessary for the urea cycle. So bicarbonate is necessary for the urea cycle. So when you have a period of acidosis, you don't want to use bicarbonate for urea. You want to conserve bicarbonate and use it to help to buffer the acidic environment. All right, so sources of your nitrogens for your urea cycle are through deamination reactions. And keep in mind that these are reversible. You have many amino acids that are simply going to release as they get broken down, converted, are going to release the nitrogen. And the liver is going to use all of these nitrogens to make urea. So in a cell, the most favored is going to be your ammonium ion, not ammonia, because of the pKa for the conversion is 9.3. And so your cells typically will stay in the ammonium ion. Excuse me. Okay, so again, ammonia, which is toxic, will diffuse across membranes. The ammonium ion, of course, is not going to. It's charged. It cannot cross the membrane. Okay, so what is a transamination reaction and what's going on though? So the purpose of transamination reaction is to collect amino groups from different amino acids and in the form of L-glutamate. So glutamate is going to then function as an amino group donor for biosynthetic pathways or excretion pathways. So the enzymes that participate in transamination reactions are named for the amino group donor. If, for example, in a alanine aminotransferase, or ALT, which is an enzyme in the liver, your amino group donor is alanine. And then you also have aspartate aminotransferase, or AST, which is also in the liver. The amino group donor is, of course, going to be aspartate. ALT and AST are serum aminotransferases, and these are clinical markers of tissue damage. So when your liver is damaged, you increase ALT and AST. Okay, so these transamination reactions are going to use pyridoxal phosphate as their cofactor. This is vitamin B6, of course, and it comes from, here's vitamin B6. And pyridoxal phosphate can go in both directions. So it's reversible. The equilibrium constant is about 1. So the species exist about equal to each other. Now, nitrogen is going to be transferred as an amino group from the original amino acid to some sort of alpha-ketoglutarate, and this is going to form glutamate, which is shown down here. The original amino acid is converted to its corresponding alpha-keto acid. So as a recap, amino acids, except for lysine and threonine, undergo transamination reactions. Enzymes are going to be those transaminases or aminotransferases. You're going to have PLP, vitamin B6, as the cofactor. It's going to be completely reversible. These reactions are going to have an alpha-ketoglutarate and a glutamate as one of the alpha-keto amino acid pairs. And they're going to be involved in amino acid degradation, moving around nitrogens, you can think of it this way, as well as synthesis, again, moving around nitrogens. One is going to be building, one is going to be breaking things down. So alanine transaminase is important for your muscle carbons and your nitrogen and being able to deliver those nitrogens from your muscle to your liver. And so this is going to occur in the fed state when the muscle is building up its stored energy. And it occurs in the fasting state when the muscle is using this carbon skeletons of amino acids for energy. Okay, and so in general, you have your muscle, and your muscle is going to, for example, in a fed state, pick up blood glucose, and the glucose is going to go to pyruvate, and then pyruvate is going to have a transamination reaction with glutamate and generate alanine. Alanine is going to go back into the blood, enter the liver. Alanine is going to undergo a transamination reaction with alpha-ketoglutarate and produce glutamate. And then glutamate is going to donate the nitrogen for the urea cycle. And this cycle continues because alanine is a precursor of pyruvate, which can become glucose, and you get this glucose-alanine cycle. Now, the urea cycle, also known as the Krebs-Henseleit cycle, is where the amount of nitrogen, you have to maintain nitrogen balance. So it maintains nitrogen balance, where the amount of nitrogen ingested is going to equal the amount excreted. And this is major excretion is through urea. The urea cycle is going to be mostly in the liver, and it again is there to prevent the formation of ammonia. It doesn't want to build up nitrogen to generate ammonia, which is toxic. Nitrogen travels in the blood attached to amino acids, mostly alanine and glutamine. But in theory, it can be any amino acid. And then 80% of nitrogen waste is excreted by the liver urea cycle. Okay? All right, so let's talk about the urea cycle. So the urea cycle is going to begin in the liver mitochondria, and then it's going to be completed in the cytosol of the hepatocyte. And ammonia is going to enter the cycle through that ammonia ion, shown here. And so you have, again, glutamine, and this is going to come from other tissues. And glutamine will enter the mitochondria through glutaminase, become glutamate. And you can also have glutamate through transamination reactions enter, become glutamate. Glutamate dehydrogenase will generate alpha-ketoglutarate and release a nitrogen. And then this nitrogen is going to be the first nitrogen that enters the urea cycle. And the first step is this carbamoyl phosphate synthase 1. So this is going to use bicarbonate, ATP, in this reaction and form the carbamoyl phosphate, which has a nitrogen attached to it. So the urea cycle basically is where you're going to have your ammonium ion, and it's going to condense again with bicarbonate, and this is eventually going to convert it to urea. Now another nitrogen has to be added, and that's going to come from aspartate, which we'll talk about. So the first step is a condensation in the mitochondria, and this condensation is again your NH4+. And then this is going to be by this CPS1. And you're going to get Carbamoyl 1. Now, the next part of this is the formation of ornithine. And this is going to be arginine. And this is through arginase. Now, what exactly is ornithine? Ornithine is an amino acid, but we do not make ornithine. We do not have the genetic codon for ornithine, so ornithine does not get incorporated into our proteins. Ornithine is regenerated during the urea cycle and also can be synthesized de novo via arginine and arginase. So this is an example of ornithine through ornithine aminotransferase, and it can be generated through glutamate and a glutamate semialdehyde. Now, Carbamoyl synthase 1, or CPS1, has two different versions. It has the mitochondria form, and this is going to be the form that makes the carbamoyl phosphate. And this is that two-step reaction. This is going to be regulated. So it's a positive regulation. By what's called an obligate activator. And this obligate activator is N-acetylglutamate. So remember, whenever there's nitrogen around, your body has to deal with that nitrogen. And so this obligate activator makes sure that if there's nitrogen around, this urea cycle moves forward. So that's why it's an obligate activator. So you have acetyl-CoA plus glutamate and N-acetylglutamate is made, this is called NAG. And this enzyme that makes NAG is N-acetylglutamate synthase. Or NAGs. Now in the cytosol, you also have a carbamoyl synthase, which is going to be involved in pyrimidine nucleotide synthesis. And this one has no obligate activator. So it's very different. Okay, so reactions of the urea cycle. So we have, again, in our mitochondria, we have our bicarb, we have our nitrogen. They're going to combine using water, of course, carbamoyl phosphate synthase 1, CPS1, and they're going to generate the carbamoyl phosphate that we've talked about already. And then ornithine from the cytosol is going to enter the mitochondria, and it does a condensation reaction with carbamoyl phosphate. And this is through an ornithine transcarbamylase. And then it's going to generate citrulline. Citrulline is then going to leave the mitochondria. So ornithine comes in, so your citrulline will leave mitochondria as your ornithine will enter the mitochondria for a 1-to-1. So this is a 1-to-1 ratio, and they have a translocase. And of course, that's on the inner membrane of the mitochondria. Now, aspartate is going to react with your citrulline and donate a nitrogen for urea formation. So remember, we generated citrulline from our carbamoyl phosphate plus the ornithine. Citrulline exited, is now in the cytosol. A nitrogen from aspartate will be added to citrulline. This is going to require ATP. This is an arginosuccinate synthase that does this reaction, and it generates arginosuccinate. Now, arginosuccinate is then going to form fumarate, and arginine through an arginosuccinate lyase, so it gets cleaved. Arginine is then going to form urea through arginase and recycle ornithine, and the cycle can keep going over and over and over again. And you can see that your fumarate is made. So your fumarate can form malate. How should I do this? And fumarate can also form oxaloacetate. So this looks like an OR. Okay, so regulation of urea cycle is going through allosteric regulation. And again, this is a feed forward because the goal is to eliminate excess nitrogen. So if there is excess nitrogen, you want this thing moving forward. So when you have a high protein diet, you're going to increase the expression of the enzymes that are going to be part of the urea cycle. A low protein diet will decrease the enzymes, so it's as needed. It's similar to ethanol. Starvation is going to increase the enzymes because during starvation, you're going to want to break down your proteins for your carbons to be used as energy. So the allosteric regulation, you have arginine is going to stimulate more ornithine production. And you have arginine that's also going to stimulate the formation of NAG. Which is N-acetylglutamate. And N-acetylglutamate is going to stimulate that first enzyme. You remember, it's an obligate activator of CPS1. So here's your NAG. Okay, so NAG is an obligate activator of CPS1, which is that first enzyme in the urea cycle. Now, NAG is, again, going to be activated by arginine. It's going to be made by the N-acetylglutamate synthase. Arginine is going to be a positive regulator of this reaction. You have an acetyl-CoA plus your glutamate. It's going to generate NAG. And this is going to be an obligate activator of your carbamoyl phosphate synthetase 1. You can think of this similar to when we talked about your glycolysis, fructose 2,6-bisphosphate as an activator of PFK1. It's kind of similar. When you have too much nitrogen, your acetyl-CoA is going to combine with glutamate just simply because they're there and they're in high amount. And you're just going to push things forward because, again, arginine is going to be there in a high amount because you're releasing a lot of nitrogen from muscles and other tissues and bringing it all to the liver. And so all of this is sort of going to converge to making this obligate activator of your urea cycle. Now, urea cycle disorders, you can imagine they are going to be pretty deleterious. If you lack any enzyme, it's going to lead to death. And so usually the infant will not even be born. It'll be either before birth or shortly after birth. Deficiencies of enzymes are viable. Okay, so remember that ornithine transcarbamylase is the X-linked recessive. All the others are autosomal recessive. That's what AR is. So they're not very common. And the most common is going to be your X-linked recessive. And it's going to be, of course, more common in males than it will be in females. And so you can imagine that you know what these enzymes do. So any defect will lead to a defect in generating urea. And so if you have too many nitrogens, that will be more of a phenotype. And if you're able to control the amount of nitrogens, the diet of these individuals. Now, the disorders are going to be dangerous because NH3 is toxic. So anytime you accumulate nitrogens and not able to push it through this, you have the ability to potentially form NH3. So you need to fix nitrogen to glutamate or to glutamine or alanine. Okay. If you see an increase of ammonia in the blood, you also typically see an increase of glutamine. Because remember, glutamine can take 2 nitrogens. So it's almost like you're getting saturation of your glutamine. So you'll have an increase of glutamine, you can't make it anymore, and so now you have an increase of ammonia. The most common deficiency is that X-linked OTC deficiency. Now, with respect to the brain, you have ammonia, which we know is toxic, but ammonia itself is a very serious neurotoxin. And hyperammonemia is going to have negative effects primarily in the brain because ammonia can cross membranes. It can also cross the blood-brain barrier. Blood-brain barrier. One of the things that ammonia does is it disrupts the metabolism of your astrocytes. Now, when ammonia enters the brain from the circulation, it does get incorporated into glutamate, and it forms glutamine. And this is through that glutamine synthase. And so more nitrogen that's available, the more that's going to affect glutamate. Now, glutamate is an excitatory neurotransmitter. So in excess nitrogen, you can imagine that most of your nitrogen is going to go in this direction, like a big arrow. And so you're going to have a decrease of glutamate. And you're going to have a decrease of your excitatory neurotransmitter in excess nitrogen. Because you don't want to build up ammonia, and so you're pushing all your nitrogen to glutamine instead of keeping it at glutamate. Okay, so neurons are going to respond to the excitatory neurotransmitter glutamate. And these are glutamatic neurons. And glutamatergic neurons are going to be responsible for encoding information. They're responsible for retrieval of memory. They're going to be responsible for spatial recognition. And just in general, consciousness. So if you have excessive excitation of these receptors, you're going to lead to injury, hypoxic injury, hypoglycemia, stroke, and epilepsy. So the brain does have a glutamate-glutamine cycle. And so how this happens is that you have your capillary, and in here you will have your NH4 or NH3, and then you'll have your astrocyte. It's kind of this funny oops. And so you'll pick up nitrogen, and then your nitrogen is going to go through a few steps to generate glutamine. And so your glutamine is going to have two nitrogens. And then this is going to be released to a presynaptic nerve. And so you have glutamine. Okay. And then glutamine can go to alpha keto. And you can make energy, of course. And then glutamine can also go to glutamate. And this can be excitatory. Right? And so glutamate is going to be released. And then you're going to have different receptors on your postsynaptic nerve. And they're named different things. Not important for this. You'll probably get this in neurobiology. But anyways, what's important here is that if you have too much coming in, so if you have excess nitrogen, then what happens is you're going to push glutamate to glutamine. And you're going to not have the neurotransmitters in excess nitrogen. All right, so what is ammonia toxicity? Ammonia toxicity, we know that ammonia, NH3, is toxic. We know that it can cross the membrane. I'm sorry, not a plus. It can cross the membrane. The molecular basis of toxicity is not completely understood. It is thought that if you have ammonia toxicity, the end stage is going to be a comatose state in which you have cerebral edema. basically you get water flooding into your brain. The speculation is going to be on the potential depletion of ATP in the brain. And the exposure of the brain to high concentrations of ammonia leads to a loss of neurons. Because you're losing neurons, you're going to have an altered synapse formation, and then your cellular energy is going to potentially be impacted, which leads to the loss of ATP. Now, the fate of nitrogen is it's going to be, one, reused to make amino acids. It's going to be reused to make nitrogen-containing compounds, or excreted. And excess must be excreted. Now, both the citric acid cycle and the urea cycle are going to be linked, and this is the aspartate arginosuccinate shunt. And basically, this is linking the separate pathways in which the amino groups and the carbon skeletons of the amino acids are actually being processed. So we have the urea cycle, and in the urea cycle, again, we have ornithine binding with carbamoyl phosphate to generate citrulline, and citrulline is going to pick up a nitrogen from aspartate to generate arginosuccinate. Arginosuccinate is going to generate arginine, which you're going to get urea. Arginosuccinate also generates fumarate. Oops, let me go to a different color. Fumarate. And then fumarate can generate malate. Malate can then re-enter the mitochondria, enter the citric acid cycle. You can have oxaloacetate made. Oxaloacetate through transamination reaction can generate aspartate, and then aspartate can go out here. Aspartate, also through a transamination reaction, can generate oxaloacetate, which can become malate and re-enter this TCA cycle. So you have aspartate, which is going to be a link between the two cycles. So there is an interrelationship of this TCA cycle and urea cycle. The urea cycle reactions are in the cytoplasm except for the citrulline reaction. And just keep in mind that ornithine is not incorporated into proteins because we don't have any codons for ornithine. Okay, so these are the pictures from your book for the same thing. So again, it's this arginosuccinate, which links the urea cycle to eventually your TCA cycle. Now, in fasting, your liver has to maintain blood glucose. Right, so it needs to have carbons for gluconeogenesis. The muscle uses proteins. So it's going to release, let's break down proteins for energy. And then the nitrogen is usually going to go to alanine. And then this can go to the liver and generate pyruvate, which then can make glucose. The alanine is also going to release a nitrogen, and in the liver, this goes to the urea cycle. So in general, you have 2 alanines will equal 1 glucose and 1 urea. Okay, so this is just showing how this all happens. Actually, it's 2 glucose, sorry. All right, so keep in mind, ammonia is toxic. We want to take care of those nitrogens. Best way to do that is through the urea cycle. If it gets overwhelmed, you can lead to toxicity. One of the nitrogens is going to enter the urea cycle through carbamoyl phosphate. The second amino group is going to be from aspartate. Arginine is an immediate precursor of urea. Through arginase, you make urea and regenerate ornithine. Ornithine is going to be regenerated at every single turn of the cycle. So net result is you have ornithine to fumarate. They are both intermediates of the TCA cycle, thus they are all connected. And activity of urea cycle is feed forward. So if you have more nitrogen, you're going to generate more enzymes that are going to metabolize more urea. Less nitrogen, you're going to transcribe less enzymes, less urea produced. And so just keep that in mind. If nitrogen's present, your body has to deal with it.